I'm Jerry Tuskan. I work for the U.S. Department of Energy at Oak Ridge National Laboratory, and I'm a geneticist by training. So we began to examine the DNA of poplar through the genome project that we had 10 years ago. At that time, we discovered that there were fungi and bacteria associated with the root systems of the samples we were collecting. But at that time, the idea that there was this microbiome associated with plants and that it was predictable was not generally accepted. So what we've done in the most recent past was use genomic sequencing to isolate and identify these bacteria and fungi that are intimately associated with the root systems, or in some cases living inside the plant root systems themselves. In soil, there's tens of thousands of microbes, viruses, fungi, and bacteria, and some of those have beneficial effects on plant growth. They improve drought tolerance or increase nutrient uptake, but managing that or controlling that's difficult in this competitive environment. We began looking at the signaling molecules that a plant produces to attract its beneficial associates, its symbiotes. And we've identified proteins that are secreted into the soil that ultimately attract specific bacteria that convey phytohormone production or phosphate mineralization. And in understanding that molecular language that's used between bacteria and fungi, we are able to then control the genotypic expression from the host's point of view and improve the probability that those microbes in this diverse community in the soil will ultimately associate with the plant and therefore predict performance or productivity. And as a result of establishing those favorable relationships, the plant will grow at an accelerated rate. The plant will also have drought tolerance and the ability to take up minerals without fertilization. So we talked a little bit about the phosphorus and phosphates, and known reserves of phosphates are limited. If we could identify bacteria that mineralize phosphates more effectively in the soil, then we'd have to distribute fewer fertilizers to achieve the same amount of growth in our agronomic systems. So the human microbiome has been studied for years. It's been known for at least the last 7 years that the microbiome in a human's mouth influences tooth decay. And so by managing that or manipulating that, you can reduce the amount of tooth decay. So I have colleagues that have been studying this for some time, and they began looking at the effects of communities in our guts, in our mouths, on our skin, and began associating those variations in community structure with phenotypes or diseases. And in talking to them, we took that same idea and began looking at that in plants to see if we could predict the occurrence of a disease, or in a positive sense, enhanced growth. We think we found communities of organisms that will allow us to accelerate or enhance plant growth.